In this video, we're going to go over how the amino acid is connected to the tRNA. Now, ribosomes themselves cannot tell if the tRNA they take in has the correct amino acid linked to it. Therefore, if we want accurate translation, there's two important steps that must happen. One, the correct amino acid must be attached to the tRNA, and this is carried out by amino tRNA synthetases. Two, the correct amino acyl tRNA, that's the tRNA with the amino acid on it, has to correctly pair with the mRNA at the ribosome. So the proteins that add the amino acid to the tRNA are amino acid specific amino acyl tRNA synthetases, or AARS. They're going to add the amino acid to the 3' ribose to form our AAtRNA. And this reaction has two steps, but is carried out by a single enzyme. First, our amino acid is activated by a reaction with ATP, in which we get our amino acyl AMP in pyrophosphate. We say this is activated because this phosphate is very high energy, this amino acid phosphate group. So removing this phosphate is favorable. Then our mixed anhydride, our amino acyl AMP, will react with our tRNA to form our AAtRNA. Now, this reaction is driven by the hydrolysis of this inorganic phosphate. So we make this PPI, and this PPI is broken apart to two phosphates. So all the energy for this reaction comes from our first reaction. And so we take this ATP, remove two phosphates, attach it to our amino acid. This amino acid is now high energy, and by kicking off the last phosphate, we're going to use that energy to attach it to our tRNA. Now let's look at these amino acyl tRNA synthetases more in depth. And most cells have at least one AARS for each of the 20 amino acids, because we make proteins that have all 20 amino acids in them. Now, AARSs are actually a very diverse group of enzymes, and they all have different sizes and different quaternary structures and little sequence similarity, which is odd because these AARSs basically do the same reaction just for different amino acids. And what this tells us is that these AARSs didn't really have a common ancestor that all 20 grew out of. Now we have two general classes of AARSs. We call them Class 1 and Class 2. And Class 1 and Class 2 have these differences. Class 1 has this called the Rossmann fold. Class 2 does not. Class 1 AARSs can recognize the anticodon of the tRNA. Class 2 enzymes generally do not interact with the anticodon. Class 1s will amino acylate the 3' terminal 2' OH group. So it's the last nucleotide on the tRNA at the 3' end, and then the 2' OH sugar of that. While Class 2 will charge the 3' OH group. What's interesting about this is for protein synthesis, you need to have it on the 3' group. So the Class 1 puts on the 2', but then it gets switched over to the 3' later on. And specificity, Class 1s generally have amino acids that are larger and more hydrophobic than Class 2 does in general. So how do these AARSs know which tRNA to put their amino acids? Well, through various experiments, we have discovered that there are unique sites that AARSs use to bind to their tRNA. we call these contact sites. And our yellow circles here are showing us just a conglomeration of our contact sites, where the bigger circles mean more likely or more frequently used to be a contact site. And when we look at this map, we can see that our contact sites are clustered in the acceptor stem and the anticodon loop, and some other points on our inner surface of the L as well. Now, there's little regularity in how these synthetases recognize their tRNA, which makes sense based on the last slide in saying that they're all unique for the most part and they don't have a common ancestor. So it would make sense that there's a little regularity in how they recognize their tRNAs. And not all of the AARSs use the anticodon. Only some of them do. And these identity elements are so varied with how they interact with the AARS, they are sometimes referred to as second genetic code that's currently trying to be cracked of how do the tRNA and AARSs interact with each other. So we're going to look at an example of class 1 and class 2 AARSs that have been studied so we can get a better idea on generally how they work. So what we're looking here is the class 1 GLNRS. And class 1s interact with the anticodon. So we have our enzyme here in blue that has a very elongated shape, where one end of the enzyme we interact with the amino acid portion, the acceptor stem, and the other portion we are interacting with the anticodon loop. Now, the identity element for this enzyme are all 7 bases of the anticodon loop. And for this tRNA, the bases are splayed out and they each bind to a separate pocket in the enzyme. So that's where our specificity is coming from to know that this tRNA is supposed to interact with this enzyme. Now for the 3' end, that plunges deep into our protein pocket where we also have ATP and glutamine waiting, and that's how we're going to attach our glutamine to our enzyme, by having our acceptor stem deep in the pocket. Now this is an example of our class 2 enzyme, ASP-RS, that's found in yeast. What's interesting about this enzyme is that we bind 2 tRNAs at once, symmetrically. The contact for this binding happens in the acceptor stem and in the anticodon region. And when our tRNA binds, it can bend up to as much as 20 angstroms. And the reason that we have this bend is that in our anticodon stem, we have a G30, which is base paired with a U40. Now, this is non-Watson-Crick, and so this is the hinge of our bend that's allowing this bend to happen. When you look at nearly all other TNAs at this position, you have a Watson-Crick base pair. So this specificity is allowing this tRNA to bind or bend, which allows this enzyme to recognize its correct tRNA. And we can take this information with our previous information of class 1 enzymes and determine that tRNA with a synthetase is really dictated by its 3D interactions with the protein. That is the induced fit model. You'll bind the tRNA and then bind it more tightly, and only tRNAs with the correct specific geometry or bases there will interact with our enzyme. So that's why we're not really looking for a specific codon to tell us which tRNA goes with which synthetase. We're really looking at our geometry features, and this idea of geometry of the protein, or in this case the geometry of the tRNA dictating how it interacts with the protein, should be a common thought at this time in biochemistry. When we looked at enzymes, looked at substrates, it's always been the geometry of the substrate with the enzyme that dictates that these two things should work together and dictates their chemistry. And this is why way back in Biochemistry 1, I made the statement that structure defines function. So it's not so much, well, let me back up for a second. It's not so much that we don't care about what the structure of our protein or structure of the tRNA is. It's just not as important as what does the 3D model of these biomolecules look like. Because once we have the 3D models, that's how we can really understand the chemistry of these biomolecules. And with that, that's the end of this video. I will see you in the next video.